Well, welcome everyone to this session which we're calling Rewriting Human Genes. I'm Joe Palca. A couple of pieces of housekeeping to start with. Please turn the ringer off on your cell phone. Someone suggested to me that if your cell phone goes off during this session, it means you have a burning question, and I'll ask you to come up on stage, stand in the middle, and say it out loud, and then sing your college alma mater. But we do want you to leave your phones on because you're welcome to tweet questions up to me. I have a pad here which I'll be getting them. The hashtag is Global Health. And anybody who's out there who would like to ask a question, feel free to tweet them at hashtag Global Health. I have a pad here that will receive them. And that's really about it. We're going to have a 45-minute discussion about genetics, after which there'll be a quiz. So please be sure and take notes. No, there's no quiz. This is just an attempt to catch up with some of the most interesting science that's being done in the world right now. And two of the people who are directly or directly involved in it are with me on the panel. First, there's Jennifer Doudna from the University of California, Berkeley. She has been very popular lately for a technology that I'm sure we'll start to talk about called CRISPR. And we'll let Craig Mello tell you exactly what that stands for. No, I'm only teasing him. We also have with us Craig Mello. He's done some interesting things in biology, including working with something called RNA interference, which is an interesting way of silencing genes. And some committee in Sweden decided that that was a very interesting piece of research and awarded him the Nobel Prize for doing it. So they have credibility. I'm not saying you should believe everything they say, but they have credibility. So I thought Craig made the interesting point that rather than launch right into rewriting genes, let's do a little biology 101 and talk about what we're talking about. So Craig, what do we mean when we talk about rewriting genes? What are genes?
Okay, great. Thanks, Joe. It's great to be here and welcome everybody. I've had a lot of experience in the last few years talking to lay audiences about science, and one of the things that you realize is that our vocabularies don't mesh up very well. I've given whole lectures and then at the end had people say, well, RNA is protein. Someone has a burning question. Things like that. RNA and protein. What do those words mean? These are really jargony terms, if you will. So I'm going to just give you a very quick, brief introduction to gene expression. So gene expression is really, really simple. There's an alphabet that has 4 different letters. Just 4 letters in the entire alphabet. And there are basically 20 different words that can be spelled in a series of, what are 3-letter words can spell each of these different amino acids, which make up the 20 different building blocks of protein. So you have in every cell your genetic material, your DNA, your genome, which is billions of nucleotides long. The nucleotides are the letters. And wherever there's a G nucleotide, it pairs up with a C. Wherever there's an A, it pairs up with a T. So those are your 4 things, and that's basically the structure that forms into this beautiful spiral double helical structure and explains so much about, for example, how your DNA replicates, because when your DNA unwinds, each strand can encode through this very precise mechanism of base pairing, can encode for the other strand. So you always can template a new strand using either of the 2 strands of your genome. It's really simple stuff. It's really basic. And then when your DNA genes are expressed, it's transcribed into RNA, which is another 4-letter type of nucleic acid. It's another form of the genetic code. It gets processed into what we call messenger RNAs. Then those are decoded by reading the sequence of nucleotides in the genetic code 3 at a time. And each of the 3 letters of code specify a different amino acid that's then assembled by this amazing machine called the ribosome into proteins by assembling the amino acids that are called out, the 20 different amino acids, which have all these different chemical properties, are then assembled into a linear chain that make your proteins. So gene expression is incredibly simple. Everybody should know this. It's a starting point for this discussion because your genome is made up of this organization of letters that specify all the different types of proteins that your body can make. So that's my sort of Bio 101 introduction, and we can sort of spring off from there.
And now I'm going to repeat it to make sure that I've got it right. But also to say one of the things that boggles my mind always is that you can, from these 4 letters that combine in go in various orders, it describes everything. I mean, everything in a living human being, and I think that's kind of an amazing concept. But transcribe, I just want to make sure, and express, those are 2 words, and protein, those are 3 words that have different meanings in a non-technical sense. So I just want to say transcribe is sort of just reading the code, right? Something that comes along and says, oh, here's what this is. And then express means to build the protein or turn it on, essentially.
Yeah, I think, again, I lapsed into jargon. So transcribe is just copying the DNA into RNA.
Right.
And then where the code gets read is by the ribosome, which reads each 3-letter word, assembling a different amino acid to match each of those as it goes along the mRNA sequence.
Right. And finally, the protein is a word. So protein is also, it's not the meat that you get in fish, the stuff in fish. No, but it's serious. It's one of those words. It is, but it's one of those words that scientists use in a slightly different way than the public thinks about it. Proteins are the stuff that does the things inside the cells inside our body. So if you want to change, if you want to make insulin, right? Insulin is a protein. So you have to have a gene that has the instructions to make insulin. And the cell has the machinery to read the instructions in the genome and transcribe them to turn them into a protein that makes insulin that does something in our cells. Okay? Okay, it went by fast. See, that's what happens. You get into, this is we have to start here because otherwise the rest of this talk isn't going to make any sense. You have to understand that the genes have the instructions to make things. They can either make them properly, which is health and good balance, or they can make them improperly, in which case something bad happens. It could be cancer, it could be diabetes, it could be neurodegenerative disease, it could be a lot of things. But just keep in mind that the genes make the proteins which do things. Okay? And the steps are classically DNA, RNA, protein. So if you ever get confused, those are that's the basics, right? DNA, she's nodding, so I feel good. DNA, RNA, protein, instructions, transcribe into proteins that do things. Okay. Whew. Jennifer, what part of that world do you focus on in your research?
Well, my lab has always worked on RNA, so trying to understand the function of this molecule that a lot of people thought for a long time was less interesting than DNA or proteins. It's kind of the molecule in the middle. And so we've always had an interest in understanding what it does. And I think one of the exciting things that's happened in biology, and certainly Craig can speak to this over the last, well, really couple of decades, is that there's been kind of this appreciation of the incredible diversity of RNA molecules and the importance of their function in cells and really for human health.
Right, and one of the keys that we're going to get to is that you can change the DNA and that does something to the protein, but if you do something in the middle step, that also can do something to the protein. Mm-hmm. So maybe, Craig, you could talk about where your work fits in this world.
Sure, and just a couple of more points that I think are relevant. One is that you're not really an organism. Your body is a colony. Right, so you've heard about the human genome. Well, there's this microbiome that lives in you, and you can't live without it. You would not be here without your flora and fauna that are part of you. It's really amazing. So information in the form of nucleic acid sequences, such as DNA and RNA, is moving around in the environment. That's what viruses are. Bacteria have DNA in them. They're constantly exchanging information. There's benefits and there are costs. When you get infected, you can get sick. But believe it or not, there's a lot of evidence that viruses can actually help organisms sometimes. So information can move and be a positive thing sometimes, but a lot of times, of course, it just makes you sick and it's bad because it'll take over, the viruses will take over the machinery inside your cells and kill you. Now, cells have evolved mechanisms to combat or to control nucleic acids that are inside them or infecting them, both. And one of the ways they do that is they use proteins that serve as search engines. Just like you would search on the World Wide Web, they have proteins that use a short query made out of RNA, nucleic acid sequence information, is used to search for matching information in the genome. And because the four-letter alphabet is so simple, right? If you have an RNA guide molecule, we call it, or query molecule that's loaded onto one of these search engines, it will then go through the genetic sequences inside the cell and look for matching information where the G's, A's, T's, and C's all line up perfectly, so that when the guide sequence matches up, it'll form this helical structure with the target. And when that happens, the cell knows that it's got a perfect match. It's found a perfect match target. So back in 1998, Andrew Fire and I discovered what turned out to be an artificial way of entering queries into these search engines that exist in every one of our cells. And we wouldn't be here without them. It's like, imagine you had the World Wide Web, but you couldn't search it. What good would it be? Mm-hmm. Cells figured this out billions of years ago, that they needed to have really rapid ways of searching through and handling information inside them. So they use these search engines to do all kinds of things. They destroy viruses with them. They control their own gene expression with them. And then we discovered a way to make an artificial query that we could then give to the cell, and it would enter into this searching machinery. And that's very powerful because that allows us to use the cell's own searching mechanism to carry out gene regulation. And by doing this, we called it RNA interference. We could block the expression of any gene by interfering with the translation of the messenger RNA into protein. We could destroy the messenger or kill the messenger. It turns out that this has turned out to be a very useful approach for both studying genetics and biology, and also there are therapeutics that are now in clinical trials in humans. So it's an exciting field.
But the interesting thing is, this is not rewriting genes.
Right.
That's
we're talking about the RNA level. We're intervening after the DNA has been copied into RNA.
Right. So remember, you can change the code and change what the protein does, or you can do some to that middle guy and have that middle guy do something that changes what the protein does, or how it works, or whether it switches on. This is a funny concept too, because if you think about it, all the cells in our body have all the, let's leave lots of germ cells. All the cells in our body have all the instructions for all the things that we need to stay alive. But the cells in your liver aren't using the same proteins as the cells in your brain or the cells in your skin. So some of those genes have to be turned on or expressed, while others are not. And so if you suddenly find that there's something in the liver that's turning on that shouldn't be, or that you want to turn off this thing to see what it does to make the liver function properly, well, you come along with Craig's probe and you send it into the cell and it goes and turns that gene off, and then you say, oh, I wonder what that did, and you look and see. And that's sort of how the research works, if I've got it right. Mm-hmm. But Jennifer, you're actually doing something a little different than that, because your lab has come on to this, well, maybe actually it's a version of that. It's not quite that.
Yeah, I guess you could call it a version of that. I mean, I think one of the interesting things when when Andy Fire and Craig Mello made this discovery about RNA interference, one of the curiosities, I guess, at the time was that that bacteria, which is a very, as you know, many different types of bacteria on the planet, none of them seemed to do this. And people could not find the molecular machinery for RNA interference in those types of cells. And so the question that we had was, we were sort of interested in how these RNA molecules do this searching, and we were studying that. And then in around 2005, there were a few sort of obscure observations about bacterial systems that looked like they might be aligned or similar, at least, to RNA interference. There was no experimental data for this at the time. It was based on looking at the genomes of bacteria and realizing that many bacteria have a way of stealing little pieces of DNA from viruses that they get infected with. They integrate or they incorporate those pieces of DNA into their genomic or genetic material, and then they make an RNA copy of that information. And just like Craig described, they use those pieces of information in the form of RNA to search the cell for matching sequences that might come from a virus. And if they find a match, then they have proteins that can engage with that, at that point, that piece of DNA that has been identified as foreign, and lead to its destruction. And so that was a hypothesis, and we started investigating it, thinking that it would be very interesting to just, we were just curious. Basically wanted to understand, if bacteria really were capable of that sort of activity. And, you can, if you think about it, it's really like an acquired immune system, right? You acquire information from your invader, and then you use that in the form of RNA to protect the cell. So that was the idea that we set out to test. And so in the course of doing that research, and of course other labs started to investigate this as well, and over the next few years it emerged that in fact this is exactly what happens in bacteria. They have this ability to acquire sequence information and then use it in the form of RNA, so a very interesting parallel, I would say, to RNAi. And but I'm a biochemist and a structural biologist, so I'm very interested in the molecules that do this. How do they work? And that's the question that we really wanted to address, that led to a technology that is now really taking off for genome engineering.
So explain why. So people have been manipulating genes and doing things with genes and rewriting genes, and there's genetically modified organisms and there's all this stuff. But yet what you've done, this new idea, this thing, it's called CRISPR, which again, we won't tell you the acronym because we'll both, somebody will get it wrong and then somebody else will laugh and it'll be embarrassing. But CRISPR allows you to do something that's different, even though it's doing some of the same things. So what's different?
Well, I guess I would say what's different is that, I guess the way that I think about it is it's like a molecular scalpel. It's a very precise tool that can be used to identify a particular place in a genome. And you think about, the vastness of the human genome, for example, and imagine knowing that there's a single change in the four-letter sequence of the genetic code in a human cell that is giving rise to a defective protein. I could, many examples of this. Cystic fibrosis is an example. Sickle cell anemia is another example. Lots of genetic diseases that are caused by a known mutation or change in the DNA that creates a bad or ineffective protein. And so what if you had a precision tool, like a scalpel, to go in and actually make the change, correct that exact site in the DNA and not affect anything else in the DNA? Very, very powerful. And that's basically what this CRISPR technology enables, is that kind of precision change to the DNA.
Right. Can I jump
Yeah,
in
sure,
for a minute?
please.
Yeah.
I mean, Jennifer's lab has been working again on the structures of these proteins that do this. So in RNA interference, the search engine searches through the messages that are out in the cytoplasm and sometimes in the nucleus as well, but it's basically searching single-stranded sequences for matching information. But this enzyme, this bacterial enzyme, and this is really basic science, because when Jennifer and others started working on this, there was no glimmer, really, that this would actually be useful in humans or in human cells. But basically what this enzyme does is it searches the same, sort of the same way, in a similar way, for perfect base pairing interactions, but it opens up the DNA, it unwinds the DNA, and it searches both strands of the whole genome. Billions of nucleotides of information. It searches through all that information, somehow scanning the whole genome for a match. When it finds a precise match, it actually allows this base pairing interaction that's highly specific, and then it cuts not only the strand that's base paired to, but the other strand as well. It has two enzymes. It's so beautiful, and the structures that Jennifer's lab has made are just mind-blowing, showing how this enzyme can engage the DNA and make these very precise cuts. So it is just like Jennifer said, like a scalpel that you can direct towards any part of the genome to make these extremely precise cuts.
But that's a very important point. So something that to appreciate about this that helps you understand I think the power of this technology is that we figured out how to program this. Okay, so it's programmable. So you take the ability to program this scalpel and you combine that with the genetic information that we have for the human genome. We know the sequence of many human genomes now, but other genomes from other organisms as well. And so you have that information, and you can now program this CRISPR tool, it's a protein called Cas9, to actually recognize a site that you know is defective in the genome to make a break in the DNA. And then after that occurs, the cell can repair that break by incorporating new genetic information at the site. And there are various ways to control how that repair happens. And so you put all of those technologies and information together, and it creates an incredible opportunity in biology. And this is what we're seeing right now. It's just an incredible explosion of research that's using genomic information from humans, but also from other kinds of organisms, combined with the technology to now make changes, very precise changes in that information, and do it quickly, and also, very importantly, do it simply, right? So maybe it sounds complicated, but it turns out that for anybody that has a basic knowledge of molecular biology, this can be done easily and quickly and effectively. So we've had high school students come to the lab, and within a few weeks of training, they're doing this type of precision genome editing.
And I was just thinking, one final analogy that may help a little bit in kind of getting your head around this is it's a little bit like, first we had the book, or like think of it as a word processor. First we could write all these words down, and then we had this function that was allow you to find, so you could search for words, and then the next step was search and replace, or search and, so you get that extra step of not only finding things and maybe taking them out, but now you can find them, take them out, or put something new in a very powerful way. And so as long as you know the book and you know the instructions to use the word processor, you're good to go, sort of. Now, Craig, you started out by talking about diseases, human diseases that might be, and that there are already therapeutics that are based on the RNA interference technique. And when we first were kicking around ideas for how to name this session, we were thinking about, okay, the end of genetic diseases. And that made you uncomfortable. So I want to talk about why that is. I mean, where are we realistically in terms of applying these tools into something that people say, oh, well, that's great. I'm glad you discovered that. You know, my aunt is alive because of it.
That's a great question, Joe. And to answer it, I'll just sort of go back to last night. We had a discussion at dinner about, what are the major gaps in knowledge that we face. And I made the analogy of my old friend Neil Shubin, who went up to Canada and high Canadian Arctic and found this amazing fossil of a fish from 350 million some years ago that had these vestigial forelimbs. And when he came back and everybody was celebrating, his colleagues congratulated him for creating 2 new gaps in the fossil record. But in genetics, that's exactly what's happened with genome sequencing. So when you sequence a genome like the human genome, it create a great deal of new knowledge, right? But as with all science, all new knowledge actually creates more questions than answers. And so with human genetics, now we have very personal gaps in knowledge because we as individuals may know that we have a genetic, we have a lesion or a polymorphism in a gene that may be linked to a disease, something terrible like Alzheimer's or a neurodegenerative disease like Huntington's disease. And yet, knowing what's wrong with your DNA doesn't mean that we have a therapy, doesn't mean that we can fix it. And increasingly now, when you go to get a health checkup in the hospital, or if you unfortunately get cancer or someone in your family does, then all of a sudden you have this additional burden of new information, right? Your dad might have cancer, and then you start to wonder, well, if he has the gene, do I have the gene that predisposed him to that cancer? My dad had prostate cancer, so right now there's no genetic test for that. But these are the kinds of burdens and gaps in our knowledge that are affecting all of us now when it comes to healthcare. So genetics in humans is now driving laboratory science because we can take a genetic lesion that's known to be linked to disease in a human, and using CRISPR, we can model that disease now in an organism where we can study it much more effectively. A fruit fly or a worm or a mouse, we can now incorporate exactly the same lesion that causes a disease in a human and then study it in an animal. And that's very powerful. So probably more important than actually developing a therapy based on fixing the human disorder, we can now study in an animal setting where we might be able to find a small molecule or some other biological approach, an antibody or something that would have a therapeutic effect for that patient. So the era of genetics and human genetics is absolutely upon us. There are potentials for applying this technology to directly correct human genetic disorders in human cells. So there's that, but there's also this reverse flow where we used to model or try to understand biology in a model organism and then try to extrapolate from the human. Increasingly, we're taking information from the human, genetic information from the human, and now trying to model that in organisms. And so there's a huge need for additional research because there are thousands of human disease-related genes now. And it's a really exciting time right now in genetics.
Just to remind people, the hashtag Global Health, if you have a question that you want me to see on this iPad, or if you're out in the World Wide Web land and you want to ask questions, please do. But it brings me to a question, Jennifer. 30 years ago, probably, just about, when we had the gene that causes cystic fibrosis, we knew what it looked like, and we knew some of the places where it went wrong and could cause disease, because that's another problem with genetics. They gave names for genes that are necessary and make people think they're bad. The cystic fibrosis gene is a gene we all have to have for our cells to work properly. It's only when that gene is incorrect that we get the disease. Bad genetics. If they had asked me, I would have stopped them from using that terminology a long time ago. Anyway, they do, it doesn't matter. But that was in a day, 30 years ago, when we had gene therapy. Oh, we know the sequence of the gene, we can make a proper copy and scoot it back in 30 years ago. And there is no gene therapy. I mean, there's the beginnings of gene therapy in some places. I'm not saying. What's different? Why is CRISPR, are we going to say 30 years ago we thought, well, CRISPR was going to give us the answer of how to fix these genes, but now we don't? I mean, what's different, do you think?
I think 30 years ago, to do an experiment like you're describing would require using, for example, a virus to deliver a normal copy of the gene. And the way that the virus works is to go, it gets into a cell using a sort of an infection pathway. And then depending on the type of virus, either it integrates into the genome, but it integrates where it wants to integrate, not where you want it to go, or it stays inside the cell but doesn't actually replicate or get copied into the genome of the cell. So yes, you could introduce the normal copy of the gene using those sorts of approaches, but it was very difficult to control the way that gene would then be expressed or would be used to produce the correct copy of the protein in those cells. I think the difference now is that with a precision tool, the CRISPR system allows scientists to, if you know, and we do in this case, where that gene is located and you know the sequence of the gene and you actually know the sequence of the mutation in the gene, you can design this system to go in, recognize it, cut it out, and then allow the cell to repair it with a normal copy of the gene. And now you have, in principle at least, you have now a corrected genomic sequence that can allow the cell to function normally.
So Craig, do you think CRISPR or that technology is going to accelerate the path to therapies? It's certainly going to accelerate knowledge about how cells work and allow scientists to do interesting experiments. And what do you think about the therapeutic potential?
I think there's a great therapeutic potential especially for cells that can be taken from patients and manipulated in the laboratory. So if you can take a stem cell population from a patient that has a genetic disorder and repair the genetic lesion in the laboratory where it's safe and you can make sure that you've affected the change you want and made no others, then you can reintroduce those cells in a setting where they'll then provide that function now to the person. Where it's harder to do is to take someone, let's say, who has a muscular dystrophy or Down syndrome or something like that and actually try to affect a change globally throughout the body of the individual or throughout all the muscles.
Or in a tissue type,
or in
right?
the brain,
Yeah.
So there it's very difficult. Again, the delivery problem is the real problem. And another thing that, I think one reason everyone has to know about CRISPR is you can rewrite the human germline with CRISPR very
Tell them
easily.
Tell them what the germline is.
Germline, that's the sperm in the egg lineage. So it used to be, really hard to do that kind of manipulation on an animal like us, but now people have done CRISPR on other primates already, on lots of different types of livestock, cows, pigs. People are doing this. And it's really a powerful tool because breeders, when they used to breed animals together, would have to cross them together and then segregate all the traits away. But now with CRISPR, you can move one trait at a time very rapidly.
Or multiple traits at a time.
Or multiple traits at a time. Yeah, it's really powerful tool for changing the genomes of organisms permanently by affecting what's called the germline. And so that's something that everybody really needs to sort of understand because, first of all, there are ethical implications there, a lot of them. And it's something that's much more likely to happen now that it's so easy to do. So it's important that people be aware that that is something that's
Before we leave that, Jennifer, can you sort of sketch out, I know you've been thinking about this, what are some of the ethical implications of modifying the sperm or egg of an organism?
Well, I think Craig, sort of sketched, the fact that this is now something that it's a technology that's easy to employ to do this. The challenge is that, yes, we have genomic information, but we don't understand, or at least I don't understand all of that information. We don't know what will really happen if you make a change at one place in the genome. Does it really only affect that one protein that gets encoded there if it's a change that's made in a gene, or are there other effects that we can't predict later on, right, that might happen? So I think that that's a really important challenge. And then I think that the question always comes up, if this type of thing were to happen with humans, where do you draw a line, and if you do, where do you draw a line? I mean, if it's okay to correct the mutation that causes cystic fibrosis, but is it okay for someone to say, well, I'd like my child to have blue eyes and not brown, right? And you can go on from there. So I think that, it's a very important conversation that needs to happen. And one of the things that I would like to do, and we have a new initiative at UC Berkeley and UC San Francisco called the Innovative Genomics Initiative. And one of our goals is to educate people about this technology and also to really try to foster the conversation about bioethics that comes up around this.
We do have a question from the internet or from the Twitterverse. This is from someone who says he's in Indonesia, or I think it's he. How can we cure cancer with genetic engineering? Is it a possibility to cure cancer, Craig?
Well, there are lots of different therapies for cancer that are extremely successful. The war on cancer, the scientists sort of always sort of overpromise, I guess, or get misinterpreted. Enthusiasm gets misinterpreted. Like the title that was proposed for this session, "The End of Genetic Disease." It's really just the beginning of understanding genetic disease, not even close to the end. But there are therapies for cancer now that are very effective, and there will be more therapies. Some of them probably will employ CRISPR and genome editing. There's a new type of therapy for cancer called immunotherapy, which I think is absolutely beautiful, where a patient's own immune system is essentially used or adapted so that it will attack the cancer. And then you can reinfuse the cells, modify the cells so that they'll recognize the cancer cells, and they'll go and then kill the cancer cells. So your immune system that normally fights and destroys infected cells can be educated so that it will attack cancer cells specifically. And there's an opportunity to use CRISPR to help make that type of attack even more precise, so that you can try to engineer the immune system to do this. And some of the clinical trials of this new therapy look very promising, where individuals, for example, with inoperable brain tumors are showing what looks like complete remission without need of a surgery. So the cells go into the brain, find the tumors, and destroy them. So yeah, there's some really exciting applications in that area. That's the one that comes to mind, but I'm not sure that you'd really be able to use this to engineer the cancer in order to kill itself, and it's probably not going to work because evolution is working against you there.
Right, but there's an interesting point also to make here, and that's the difference between a genetic disease in the sense that it's inherited and a genetic disease in the sense that something's gone wrong with the genetic machinery inside of a cell. And that's really what's happening a lot of the times in cancer. It's not that necessarily that you inherited something bad that's going to cause cancer. It could be something that changes over the course of a lifetime in the way the genes are functioning inside of a cell that can cause the cancer. Do I have that right?
Yeah, you do, yeah.
And so does CRISPR, once you make a change with CRISPR, like if you took out cells from me, these maybe bone marrow stem cells that Craig was talking about, like they do bone marrow transplants now, and you made some change in those, would that change persist if you put them back in me, and would they keep having that over and over again as they populated in my body?
Yes, if they are what are called stem cells, cells that can proliferate over many generations in the body, right?
One example of a therapy like that is the therapy to create, for people with HIV, to create resistance to HIV by mutating the CCR5 receptor on the cells that the HIV needs to get into the cell. So if you take a patient that's already infected, you get bone marrow cells from the patient that are not infected cells, make that correction, put those cells back, then those cells will not be able to get infected. So the patient will then, those cells are now under positive selective pressure because the other cells are being destroyed by the virus, and they take up residence, populate the immune system of that individual, and then hopefully you've affected a therapy. So we already know that people who have that mutation are resistant to HIV. So there are clinical trials for doing that using another version of a genome editing tool.
Jennifer, you alluded to this earlier, but I want to sort of hammer home this point that when you were doing your research over the last couple of decades, you weren't sitting in your lab thinking, okay, how can I cure human diseases? It was sort of like, oh, that would be a nice consequence, but you were asking a very, very kind of basic question about how cell machinery works. What's the connection there? I mean, how do scientists both work on something basic but have their minds open to something that might have a therapeutic potential?
Well, I think a lot of the important technologies that have come out for manipulating DNA in particular, but other things as well, have really come from this sort of curiosity-driven research by scientists who are, you know, not going after a new technology in particular, but are just investigating how things work. And in the course of doing so, they stumble across something or realize that something can be harnessed for as a technology. And that's, you know, there are many interesting examples of this. Actually, a lot of them have come from bacteria, you know. So something called the polymerase chain reaction, which is a way that we can amplify or make many, many copies of just a particular segment of DNA. That's something that came from the bacterial world, and again, was from curiosity-driven research, not necessarily to create a technology. And you know, something called green fluorescent protein, which is used for labeling cells, another idea that came from just wanting to understand, you know, how do certain types of organisms are able to turn their bodies green, fluorescent green, from jellyfish, right? So, you know, just very, very interesting. So I think to me, this really underscores the value of doing that kind of research. And I mean, Craig also, his laboratory also does research that I would say is of a very fundamental nature, trying to understand the basics of biology. And again, you know, his research came across something where, you know, they realized in the course of doing their experiments that this could actually be really interesting and really exciting technology.
Yeah, you know, the basic science really is so important and so powerful in, you know, when you're trying to understand something extremely fundamental about how cells work. And we still have a lot that we don't understand. But the thing that people don't realize is that the genetic code is the same in bacteria and humans. The same 4 letters, the same 3-letter words, and the same amino acids assembling the protein. So you can learn a huge amount. In fact, the genetic code was deciphered using bacteria and bacteriophage, bacteria viruses, to study the human, you know, to figure out the genetic code. And later on, we realized that it's the same in humans. So there are really, really fundamental questions that we need to answer. And like Jennifer's field of protein structure, one of the huge unknowns right now is how from a primary sequence of amino acids, could you or how can you predict how a protein will fold? Because there's all kinds of, even though there's only 20 different amino acids, 20 different words, if you will, they have a really intricate chemistry associated with each of them, and they assemble in 3 dimensions. So you get these emergent properties when proteins fold, and we don't understand that. Even the fastest computers we have are really lousy at predicting structure. So you have to actually determine the structure of these proteins. You can't just read the sequence and say, oh, I know what that protein would look like.
Yeah.
So.
And I think one of the best ways to think about the importance of basic research is if you think about the work that was being done in the 1950s on trying to understand what to do about polio. There were a lot of people who were designing better iron lungs because that was a really concrete thing you could do to help polio victims survive longer. But the real breakthrough in polio came from a basic understanding of the virus that caused polio, and suddenly, instead of building better iron lungs, we were designing a vaccine that prevented polio in the first place. So you sometimes say, well, what's the connection between basic research and where does it get us? And the answer is, it takes us, it's almost this quantum step forward that, you know, applied research sometimes only gets you small baby steps. Anyway, just saying. We have time for one quick question. It's a big question, but I wanted to take it. It's from the internet, and it says, who owns this new genetic tool? And I don't know whether you want to take that, Craig,
We're
and
the
spare
lawyers.
CRISPR. Well, but maybe you can just sketch out a little bit about the fact that there is some issue about that.
Yeah, and it's not something to tackle in a couple of minutes. But Jennifer and her coworkers filed some of the original intellectual property around how to essentially design guide RNA molecules that will enter into this search machinery, which I think is a really fundamental piece of intellectual property. But there's a lot of intellectual property that is coming along as people figure out other ways and better ways maybe of designing guide RNAs and to modify the enzyme itself. It's really, I mean, that's another thing, you know, this investment in basic science does lead to big economic impacts. RNA interference is a multi-billion-dollar industry. You can order a guide RNA for doing an RNAi experiment and have it delivered here by FedEx tomorrow if you want it. I don't know if they have same-day delivery, but seriously, you can knock out any gene in the human genome with a guide RNA from the, you know, ordered off the web. So, you know, there's a lot of intellectual property. It's still being sorted out, and there'll be a lot more. But that's good for the economy because it leads to startups. Jennifer and I are both involved in different startup companies that are commercializing this technology.
All right, well, I'm afraid we're going to have to leave it there. There will be an opportunity to comment on this session if you enjoyed it on the way out. And so we'll end simply by asking you all to give our speakers a round of applause.